Hello, my dear audience. Welcome to another episode of Let's Talk Micro. I am so happy that you are here with me. And if this is the first time tuning in to Let's Talk Micro, welcome. Happy to have you here. And thank you for choosing this podcast. If you are tuning in because you want to learn more about Aztreonam-avibactam, maybe you work with it in the lab, you want to learn more about it, you want to know maybe, as you know, it was FDA approved and you want to know when will it be released, we're going to go ahead and jump right into the episode. And as we do in Let's Talk Micro, we'll break it down and we'll talk about what is Aztreonam, what is avibactam, we'll talk about its activity against beta-lactamases. And of course, on the episode, I am joined by two guests, which are Dr. Callan Bleick and Dr. Mohammed Al Musawa. And both of them co-author a review about Aztreonam-avibactam that I'll be posting the link in the show notes. So let's go ahead and jump into the episode. Well, you know that for you, the audience, that here in Let's Talk Micro, we try to, we talk about everything micro and then, we have our own section on antimicrobials, and you in the lab, you definitely work with them, and you know about beta-lactam antibiotics and maybe you heard about Aztreonam-avibactam. So I thought it would be a good time to do like we do here in Let's Talk Micro, break things down and explain them as simple as we can. And doing research, we found this review about Aztreonam-avibactam, which was titled "The Dynamic Duo Against Multi-Drug Resistant Gram-Negative Pathogens." This was published in November of 2024 in the journal Pharmacotherapy, the Journal of Human Pharmacology and Drug Therapy. So with me today, I have two guests. One of them is Dr. Mohammed Al Musawa, and the other is Dr. Callan Bleick. Welcome to Let's Talk Micro.
Thanks for having us. We're excited to be here.
Thank you, Louis, for having us.
My pleasure. So now that the audience knows your name, maybe we can start with a little introduction as far as what you do and anything else that you want to share with them.
Yeah, I can start first, if Collins allows me. I'm a postdoctoral fellow specializing in infectious disease pharmacotherapy. I'm originally from Saudi Arabia, where I earned my Doctor of Pharmacy degree in 2015, and I later completed my PGY1 and PGY2 clinical residency in infectious diseases at King Faisal Specialist Hospital Research Center in 2020, which is one of the well-known hospitals in the Middle East. So through my previous experiences, I found myself drawn not only to clinical care aspect of infectious diseases, but also to the research side. Specifically understanding how antimicrobial therapy influence patient outcomes in real-world settings. I became especially interested in questions like how can we optimize treatment to reduce resistance? What strategies truly improve recovery and survival in high-risk patients? And so that curiosity led me to the United States, where I am now finishing up my postdoctoral research fellowship in infectious diseases. My current focus is on evaluating clinical outcomes and therapeutic strategies in the management of multi-drug resistance infections with the goal of improving patient care at both individual and population level. That's pretty much about myself.
Yeah, and I'll go after Mohammed here. It's gonna be hard to follow up with that one. So I'm Callan Bleick. I'm also a postdoc research fellow specializing in infectious diseases, pharmacokinetics and pharmacodynamics, with a focus on translational research. I completed my training at the University of Rhode Island, where I earned my bachelor's in biosciences as well as a minor in chemistry, followed by my doctor of pharmacy degree. I also recently completed my master's in immunology and microbiology at Wayne State University School of Medicine here in Detroit. During my last few years of pharmacy school, I actually had the opportunity to work with an infectious diseases research lab. And so that's where my interest sparked in translational science. And so from there, I realized I wanted to pursue postgrad training where I could eventually, bridge that gap between what we see at the bench side and how we can translate that information in treating patients at the bedside. So currently, my research focuses on optimizing antimicrobial therapies, understanding resistant mechanisms, and exploring innovative strategies like phage antibiotic combinations to better tackle multi-drug resistant infections.
Well, welcome again. It's so interesting before you jump into the episode how, as I take in this journey in the podcast and how much I learned about things and beyond the lab, and before, you know, first, learning about, ID physicians, and then later learning about ID pharmacists. And also, I had the good fortune of interacting with quite a few of them. I've been guests on the podcast, and I do like also because we share, for lack of a better word, I guess, that nerdiness. But in your case, it comes with, with the organism and the drugs, and then we do it on the bench side with the testing, and they complement each other so well, and that's what I enjoy. Learning beyond what happens, in the bench. Which that's why we're gonna start with some basics. In this community, we have any listeners from pharmacists to students, to lab techs, medical lab scientists with different degrees of knowledge about antimicrobials. So it's good to start from the very basic. So let's start with the questions. What is Aztreonam and what is avibactam?
Yeah, so I can start. So, Aztreonam is a monobactam antibiotic, and it's structurally distinct from penicillins and cephalosporins, but still part of the beta-lactam family. It specifically targets gram-negative aerobic bacteria by binding to penicillin binding protein 3, so PBP3, and it inhibits cell wall synthesis, leading to bacterial cell death. So, one of Aztreonam's unique advantages is that it's stable against metallo-beta-lactamases, also known as NBLs, and we'll get into more of that in a couple more questions here. But it's a class of beta-lactamases that can hydrolyze nearly all other beta-lactams, including carbapenems. And so, um, Aztreonam is vulnerable to other beta-lactamases, particularly the serine beta-lactamases like ESBLs and AMP-C enzymes. And so these enzymes can hydrolyze Aztreonam, rendering it ineffective, especially in multi-drug resistant organisms that can produce both NBLs and serine beta-lactamases. And then that's where avibactam comes in and plays a critical role. It's a non-beta-lactam beta-lactamase inhibitor that inhibits class A, like KPC, class C, AMP-C, and some class D beta-lactamases. However, it doesn't inhibit class B enzymes, the MBLs, which are among the most common, or which are among the most concerning resistant mechanisms globally. And so it doesn't inhibit MBLs directly, but what it does is it does protect Aztreonam from degradation by those serine enzymes, allowing Aztreonam inherent MBL stability to come into play.
And just for for the listeners, right? When we talk about class A and class B, we're talking about like the Ambler classification system for beta lactamases. So yeah, so then so avibactam basically allows Astronam to do the job. It's like, hey, I'll hold things here and I'll help out, and then you go ahead and do your thing. Um, so and you kind of touched a little bit on that, and you mentioned that that Astronam has able to you know we can when it comes to the class B has activity against them. Maybe is there anything else that you can touch on about it? Maybe the structure and why that is.
Yeah. Mohammed, I don't know if you wanted to go into the the structure a little bit. We're gonna get bounce around here. Yeah.
Yeah, yeah, I can take this one. So, Astronam has a monocyclic beta-lactam ring. It's unlike other fused ring structures of penicillin, cephalosporins, and carbapenems. This unique structures makes it resistant to hydrolysis by NBLs, as Collins explained earlier. However, it remains susceptible to SBLs and AMC beta-lactamases, which are frequently co-produced by the same organisms. So azithromycin alone is often insufficient to treat resistant infection. That's why they came the idea of combining these two drugs together to treat drug resistance infection.
Perfect. And like I was talking, before we started recording, for the audience, this review I'm gonna put it on I'm gonna put the link on the show notes because I'm gonna ask about some of the results and how it has performed. But if you want to dive into it and see more information, the link is gonna be there for you. You click on it and then just read it at your own pace. And so we'll kind of just touch some things here. So my next question is: so how has it performed in studies? And also, like, what kind of microbial spectrum of activity does it have?
Yeah, so starting from the recent published study in May 2025, early this year, with large in vitro studies published in Ovid. It looked at over 54,000 Enterobacterialis isolate collected from United States hospital between 2017 to 2022, and the results were pretty encouraging. So in its own, azithromycin had really limited activity against CRE. They found only active against about 3.7% of CREs, of where but when they added avibactam, the susceptibility jumped to 98.4%, which is a huge jump. So that's mean adding avibactam to azithromycin is actually made a difference. Even more impressive, this combo worked against all three major of classes of carbaminomases, like class A, including KPCs, or class B, which is the most important one, the metallo-beta-lactamases, including New Delhi metallo-beta-lactamases, and class D, including oxa 8, oxa 48 like enzymes. In fact, it had over 98% activity across the board, and outperforming the other agents like cefidrocol and tigecycline in this study. So another one global study, they also looked into 51,000 Enterobacterialis isolate, and they showed 99.9 susceptibility at MIC of 8 or less. And for MBL producing strain, the MIC 90 was low as 1 microgram per ml, which is um, summarizing this in vitro study is showing us azithromycin is powerful and it's much needed option in the fight against highly resistant gram-negative infections, especially those involving EPLs where we had almost nothing available before. However, I would like to highlight something important in vitro studies. We noticed it's had modest action against Pseudomonas aeruginosa that had EPL enzymes, which is interesting. They didn't notice any difference when they tested against azithromycin, and when they added avibactam, the MIC didn't change, which is interesting. As we all know that Pseudomonas aeruginosa has different mechanism of resistance, not only beta-lactamases, it includes porin mutation and efflux pumps and other mechanism of resistance. In terms of clinical trials, we have two clinical trials so far. So first one is the revisit trial, and this trial included 422 patients with different sorts of infection, including complicated intra-abdominal infections, hospital-acquired pneumonia, and ventilator-associated pneumonia, and they compared Aztreonam-avibactam to meropenem, and actually they found there is no difference in terms of clinical cure rates, like 68.4% for Aztreonam-avibactam and 60.57% for meropenem um, antibiotics. Um, the trial that we are looking forward to see the full result is the assemble trial. They have reported so far of 15 cases, um, is though small, however, give a more encouraging outcome. Where the sample trial they compared azithromycin versus the best available therapy for organisms that had MBL mutations, and they found that the clinical cure is around 41.7% for azithromycin and 0% for best available therapy, which is promising data for Aztreonam-avibactam. However, again, this study has small sample size, and we need more larger study to see how it will be, the outcome will be if we include like very representative patients. So to conclude, this is early but very encouraging results, especially for organisms with virtually no alternatives.
Okay, thank you for that. And so and maybe before I jump into my next question, and you touch on how it did against like a Pseudomonas, and you mentioned Enterobacterialis. So is there anything else that you can add about resistance when it comes to this drug?
Yeah, yeah, yep, of course. So like I meant, or I don't know if we touched upon like the microbiology, spectrum of activity of Aztreonam-avibactam, but it did demonstrate potent in vitro activity against Enterobacterales, like you mentioned, including MDR, carbapenem resistant, metallo beta-lactamase producing strains with, I believe, MIC 90 values ranging from 0.12 to 1 microgram per ml, with a 99.4% susceptibility with an MIC of less than 8. And so, across the multiple global studies, Aztreonam-avibactam consistently outperformed Aztreonam alone and against other comparators like ceftazidime, cefepime, piptazo, especially against resistant Enterobacterales. Now, as far as the resistance goes, resistance is still rare, but it's emerging, right? And so it still needs to be addressed, and we still need to remain vigilant. And N. E. coli PBP3 insertions, such as YRIK or YRN, YRIN, have been associated with elevated Aztreonam-avibactam MIC, so up to around 16 micrograms per milliliter, approaching those clinical breakpoints. Additionally, a novel SHV12 mutation has also been linked to reduced susceptibility. And so these broader surveillance studies, including the Rejuvenate study and Century, they have further revealed that there's this complexity of resistance among Enterobacterales. And so while CRE isolates remain relatively uncommon in the US, around 6.6 to 8%, MBLs and other carbapenemases like KPC, OXA48-like, and N-deobdominant, and regions such as Eastern Europe and South Asia. So, notably, as Aztreonam-avibactam, it again retains this potent activity against these resistant organisms, like I mentioned, with good MIC 90 values across all these MDR phenotypes. However, it is important to mention that it is species specific and variability does exist. So I guess I would say, to conclude, these findings really highlight the importance of continued genomic surveillance, molecular characterization, and stewardship efforts. As even, I'm sure as you know too, that low frequency resistant mechanisms could really compromise the utility of this promising treatment combination.
Indeed. And hopefully it stays, like we start, like that resistance, remains low. That's an unfortunate thing when we see like this last resort, MDRO antibiotics, that we get them out there, and then all of a sudden you start seeing the trend, and then more and more resistance. We've seen it with sulbactam, durlobactam for Acinetobacter. We see it with cefiderocol. So, hopefully just, but yeah, definitely the importance of surveillance. Um, so when it comes to, right, with treatment and the drugs, right? Sometimes you have drugs that are more intended for like a specific type of infection, and that's the behind the way that is. It's something that is more in your field than mine, that you understand that better. And um, so drugs for UTIs, for example. But when I'm reading about all these different, quote unquote, like last resort drugs, you see, okay, typically approved for, like a ventilator associated pneumonia, or, you see things like UTIs or bacteremia. So when it comes to this combination of Astron and Mivactam, is there a specific, like source that it is maybe, or infection that is intended for, or does it work like across, more like organism intended, or I don't know, maybe you can talk more about that.
Yeah, I can take this one. Um, so in Europe, they approved Aztreonam-avibactam based on the revisit trial, and as I mentioned before, revisit trial included infection from different sources of infection. So in Europe, it's approved for treating multi-drug resistance, including MBL from any source of infections. However, in the United States, it's approved only for treating complicated or UTI infection generally. So we need more data about other sources of infection to see like if the outcome would be different, especially like comparative studies. So far, in United States we can only use as FDA approved indication for UTI. However, like in real world practice, we know like we can use it for different sorts of infection, especially the pharmacokinetic and thermodynamic studies showed that the volume distribution and tissue penetration is good even for to the lung. So I guess as off-label use, we can use it to treat MDR from any source infection until we get more data or clinical study, real-world studies on Aztreonam-avibactam.
And right when it comes to a drug, and in this case, this antimicrobial, how is the overall like the safety of it, based on what you have studied and come up on reviews and what you know?
Yeah, so Aztreonam-avibactam has some strong safety profile, especially the safety profile didn't change much from Aztreonam, and we have a long history of using Aztreonam in the practice. And when added avibactam to Tottenham, the side effect didn't change much. In fact, in the revisit trial, the serious adverse event didn't differ significantly between meropenem and avibactam. It's like 19% versus 18% was almost like the same percentage of side effects. And the most common side effect is mild liver enzyme elevation, which is this known from previous study on azithromycin itself. Overall, it's well tolerated, especially when compared to older drug like more toxic option like polymyxins, and we know like CREs or MBLs. We used to use polymyxin, which it's really nephrotoxic drugs. I think when compared to avibactam, it's much safer than this agent.
Okay, and based on you mentioned like it was approved in Europe, and we know that it was approved by the FDA. And just to leave a little give a little context to the listeners, and if you do testing, I'm sure that, well, depending on what lab, what capacity it functions, but when you have tested for this combination, right, you test ceftazidime, avibactam, and and Aztreonam, so you do like a synergy testing, and as far as giving it to the patient, I imagine it has to be given in combination as well. But it was approved, so when would be available in the US?
That's a great question. I think that's what everyone is asking, right? So, like you mentioned, the FDA just recently approved Aztreonam-avibactam for complicated UTI back in March 2024. But we actually just spoke to some people, and it's actually expected to become commercially available sometime mid-June. So it's coming up here pretty soon. And hopefully, we anticipate broader indications such as HAP, VAP, and intra-abdominal infections to be added more, like Mohammed said, as clinical data becomes available.
Perfect. Okay. Yeah, no, I think sometimes, from when we started doing like in my facility, I guess from the nerdy aspect of things, it was like so cool. Like, okay, you put this strip here and then you, incubate and then you remove and add. And you see, like understanding the synergy and how they work together. And then you take this, like this NDM and then, Astronaut completely resistant. And then you do the synergy and then you see like a very low MIC. So I don't know, just I like to share this with the listeners about how nerdy we get and the things that we think look cool in what we do, but
Yeah.
right.
Yeah, I just had a quick question. How do you guys do your synergy testing? Or is it like a checkerboard method? I'm just curious to hear from your perspective.
Yeah, so we did our own like a validation, but so we put the Avicaz strip, so the ceftazine vacuum for the listeners. We put the strip and then we incubate it for 10 minutes, the gradient strip. After the 10 minutes, then we remove it, and in the same spot we put the Astronam strip, and then we read it the next day. So
Okay. Yeah, so it's like a yeah. Oh, like a cross e-test type of I guess synergy test.
for this particular combination, no, we've been doing that because lately we've been getting like a lot of requests for synergies when it comes to like daptomycin and ceftaroline and for MRSAs. But for this one, it's just actually on the same area where we have, so there's no crossing. It's just that we replace one strip with the other. So we let the avibactam concentration, which is the same throughout the strip, and the ceftazidime is the one that changes. So we let it, in the incubator for 10 minutes, so it's pressed a little bit, and then we replace the strip with the Aztreonam. And I think when I was reading the literature for the testing, it said that we had to do it like for 60 minutes, but then we did some internal validations, and 60 and 10 and 30, they work the same. So we left it at 10.
Interesting. Yeah, interesting stuff.
Yeah, no, indeed. I think when I was, because I did a presentation on micro about sulbactam durlobactam last year, and one of the things the study was comparing it against Aztreonam-avibactam, with this cefiderocol and some other combinations. So as I was preparing for that, that's when I just I saw the 60 minutes, and I asked him like, we did the validation and it works in 10 minutes and 30. So okay,
There you go.
yeah. So before we kind of start coming to the episode starts closing, and I'm going to ask you a question that I'd like to ask all the guests. Regarding this, is there anything else that you want to add, or anything else?
Yeah, so I would add something from stewardship perspective. I think it's really important to highlight that avibactam is more than just a new drug. It represents a paradigm shift in how we approach infections by highly resistant gram-negative infection. So for a long time, we had virtually no options for MPL producing organisms, or at least like we can say safe options for treating these MPL's infections. So this combination changed that, especially with cefidrocol and now upcoming new agents that have MBL beta-lactamase and beta-lactamase inhibitors that have MBL activity. So that said, we must be very judicious with its use. Stewardship will be critical to preserving its effectiveness because if we lose this, we are back to square one. So so I think we'll be very wise in using avibactam and other this even applicable for the new beta-lactamase inhibitors. Like we are now seeing increasing resistance with cefidrocol, which is it's cefidrocol like it's not happening so long in the market, but resistance has been like rising dramatically, which is very bad. Like we were so happy for cefidrocol to cover stenos, Mycobacter, and other gram-negative infections, but now we are losing it. So that's why I wanted to highlight this thing.
Yeah, no, definitely. And as someone that works on the bench, it always, it surprises me or when you start seeing all these requests and test for this drug, and then maybe you're looking at the susceptibility profile and you see, okay, should they be really asking for this right now? It's like it just, so those cases it's always good, especially if you work in a facility and you have like a director clinical microbiologist on site, refer those cases and ask them, hey, have a conversation. It's just not, it's good to in a way question these things and then get more education and say, hey, Dr. So and so, maybe should we just be testing this? Maybe you want to have a conversation with them, or something like that, because you do get them. And sometimes in a large facility where you get overwhelmed, maybe the easiest thing is just, oh, let's just test it and release that result. But it's good to take your time and analyze. That's what I like to tell this to the audience, and get familiar with, read your resources, know where they are, and get familiar with them. And then if you see anything that's off, just have that conversation. It's very important. Okay, so this question is just, I like to ask it to the guests, and we all have that organism, right? That we like it for some reason. Either it has significance because maybe we did a paper on it, or we like a mechanism. It can be a visual thing. With bacteria, maybe we like the way that it looks on agar, or the way that it smells, or anything. It can be anything. It can be a bacteria, it can be a fungi, it can be a virus. So let's start with you, Callan. What is your favorite bug?
Yeah, so this is tough. I have a lot of favorites here, and you kind of switched that up with me with I can add in a virus here. So, of course, I would have to say bacteriophages, right? They're like viruses that specifically target bacterial species. So I'd have to say phages, more broadly, right? But I guess specifically is in terms from a bug standpoint, it'd probably be daptomycin non-susceptible MRSA, just because I've worked with it, I've published on it, and I know how it kind of behaves against different phages. So I just find it really interesting, and daptomycin also, its mechanism of action is also intriguing too. So I would have to say that one. It always keeps me on my feet here in the lab.
Excellent. What about you, Mohammed?
Yeah, so I think Acinetobacter pneumoniae, hands down, it's a crop. It's usually carbapenem-resistant Acinetobacter and it's incredibly resilient. It has like many genetics mutations that can be implementing of challenges in antimicrobial resistance. So if you can beat Acinetobacter, then you are doing something right. So I would go with Acinetobacter for sure.
That is, and before I comment on that, yeah, with that daptomycin, yeah, we're always hoping, right, to get that MIC to one or less, and then it's like if we retested and it's greater than one, it's like okay, that's warrants a conversation and ask, okay, what do we do now and how do we proceed from here? But you mentioned Acinetobacter, and yes, and that's one all the way from when we're students, and I like to give it to mine as well and gram stain it. And then the thing is like, why is this thing staining purple? It's like, or sometimes we get this variability, so it keeps you sharp all the way from those plump cocobacillary forms. So it's staining, so it's good. I think if you come out of a program having a good grip on that organism, and I think you'll be okay because it keeps you on your toes all the way from the morphology and when it comes to on the plate and antimicrobial resistance. So those two are great ones. So well, thank you so much. This has been so great, and I enjoy talking to you. So thank you for taking the time to be in Let's Talk Micro.
Yeah, thanks again for having us.
We enjoyed it.
My pleasure. Well, there you have it. I hope you enjoyed the episode. Thank you for to Dr. Bleick and Dr. Al Musawa for their time, and I hope you learn more about Aztreonam-avibactam and you enjoy the episode. Well, thank you for listening, and you can find Let's Talk Micro on social media, on Instagram, TikTok, YouTube, and Blue Sky Social as Let's Talk Micro, on X as Let's Talk Micro 1, on LinkedIn as Luis Plaza, and I have an email address which is letstalkmicro@outlook.com. And you can find the podcast on any podcast platform such as Apple Podcasts, Spotify, Amazon Music, Good Pods, whatever you listen to podcasts, you can find it. So I invite you to subscribe to it, leave a review if the app allows you to do so, and download episodes. Either via social media or via email, you can leave any feedback, any review, any topic suggestions. They are always welcome and appreciated. And if you are looking to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book available on Amazon. These resources include cheat sheets, practice test games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. And these are some great resources from Dr. Timothy Gauthier, a very passionate pharmacist. As you know, the Let's Talk Micro podcast does not have any sponsors, so if you want to support the podcast in any way, you can do so via a Venmo link or via Buy Me a Coffee, KOFI, both links available on the show notes. And as you know, the Let's Talk Micro podcast is supported by the American Society for Microbiology. To learn more about microbes, visit asm.org. And that, my dear audience, is the end of this episode. I hope you enjoyed learning about Aztreonam-avibactam. As always, I enjoy sharing this information with you. If you're planning to attend Microbe this year in Los Angeles, stop by and say hi. I'll be there. I'm looking forward to meeting you and connecting with you. Stay tuned. Great things coming your way. So please, continue bringing that passion to what you do. It's so important. You do such great work. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.